Thanks, Casey. It's really a pleasure to be here. Glad for the opportunity to talk about gene therapy for rare diseases and kind of the progress that's been made and maybe what the future looks like. Now I'm going to talk generally about gene therapy for rare neurological diseases. I'm going to leave, as Casey said, we have been working on a gene therapy for SIRF1 related Leigh syndrome since 2018, but I'm going to leave it to Dr. Qinglan Ling, who's a senior postdoc in my lab, to present on that later today. So this is really trying to give a talk about where the landscape is right now for rare neurological diseases in general, sort of what's being done, what the limitations are, what the promise is, and hopefully what the future looks like. So, as Casey said, we do have a number of things that we're working on that have moved into the clinic, and we have worked with different industry partners along that process for several of these. So there's conflicts of interest related to that. So I'll start off when we're talking about gene therapy then there's a number of different ways to deliver nucleic acid into the body, but in terms of delivering to the nervous system, there's really one vector that's come to the forefront in terms of being able to achieve widespread delivery of a gene throughout the brain, and that's adeno-associated virus, abbreviated AAV. And this is basically a fairly simple parvovirus that was identified about 60 years ago as a contaminant of adenovirus stocks in the lab. And so that's where it got the name adeno-associated virus. But in reality, it's a completely different virus as adenovirus. It's actually it's in the parvovirus family, and it is not associated with disease in humans. So it's in a lot of ways, it's sort of a parasite on other viruses. And so, those features kind of inherently gave the idea that this might be a relatively safe vector as compared to other pathogenic viruses. It's got a pretty simple genome, and you can make a recombinant version of this virus where you delete all of the viral genes, and then you replace it with some DNA cargo up to about 4.5 kb that you wanted to carry. But this vector has now been used in over 300 different clinical trials, thousands of patients, across a wide spectrum of different disorders. And overall, it does have a pretty favorable safety record, although in the last few years, there have been some deaths and other adverse events that have happened in some gene therapy trials with AAV that are associated with very, very high doses delivered IV. But nonetheless, now we have a track record of over 10 years where some of these AAV products have received full regulatory approval, and I think we're up to about 5 FDA-approved AAV products that are being marketed. So in terms of delivering genes to the nervous system, there's a history of this in the clinic going back to the very early 2000s. I think Canavan disease and Parkinson's disease were some of the first gene therapy trials or applications. But it was, I think a lot of those trials had been, definitely mixed success. But there was one trial that I think opened the field that was published in 2012 for AADC deficiency, where I think it really demonstrated the potential of using gene therapy to treat a brain disease. And so this is not to go into too much detail about this disease, but it's basically the brain is missing an enzyme that's required to produce dopamine. And without dopamine, I think you can see that this is an important neurotransmitter. Patients are not able to really, transmit, many signals across the brain very well. Difficult to do any kind of purposeful movement. And so this is a very high morbidity associated with this disease, as well as early mortality. So this is one patient here, and this patient received bilateral intrathalamic injections of an AAV vector carrying the AADC gene that she was missing. And then this is the same patient at 13 months post-injection. Where across the board, I think the patients that were treated in this clinical trial saw substantial improvements, not a cure, but I think substantial improvements. And a lot of this was because of the biology of this particular disease, where you could do sort of local delivery to critical areas of the brain that require dopamine, and I think this expanded to over 20 patients in Taiwan, and then expanded to Japan, and now there's advanced trials in the U.S. and Europe, where quite a number of children have been treated with this disease. And earlier intervention is definitely correlated with better outcomes. So this is now about 10 years old, 10 years ago or so, of this demonstration of pretty meaningful, substantial efficacy for a gene, brain-directed gene therapy. But as I said, this utilized an approach where you're taking, doing stereotaxic administration, injecting the vector directly into the brain parenchyma, and that was sort of where the field was, again, kind of around 2010, and that could be done safely. It had been done across a number of clinical trials, but whenever you're doing that approach of stereotaxic administration into the brain parenchyma, you can basically only treat sort of small pockets of the brain at a time. And it's really not feasible to do enough injections throughout the whole brain to get very wide, expansive coverage. So you're really limited to more treating discrete areas of the brain. So basically the field, and my lab was heavily involved with this kind of transition, was exploring different vectors that could be, or different versions of AAV and different methods to inject this IV and have it get across the blood-brain barrier, or injecting the vector into the spinal fluid. And I should remind everybody that your spinal fluid is kind of produced in the middle of your brain. It basically circulates down your spine, back up your spine, and around the outside of your brain. So it's sort of one continuous flow of liquid, and a lot of drugs that are administered targeting the brain are injected into the spinal fluid. So just starting off with this approach, injecting intravenously, our lab and several other labs were exploring this and basically identified this one version of AAV. There's now hundreds of different capsid variants of AAV, but there's this one AAV type 9 that was found to have some ability to cross the blood-brain barrier. This is just injection, showing the overview of the results. If you inject this IV in a mouse, and then these are different sections through the front to the back of the brain, and all the dark staining here is expression of green fluorescent protein that this vector was carrying. And so you can see that there's widespread distribution throughout the entire brain from this IV approach. But the details of this are, if you inject the vector IV, you can look at what gets to the brain. But if you look across the rest of the body, you'll see that there's a certain amount that goes to the brain and the spinal cord. But then when you look at other peripheral organs, you're seeing much larger amounts of the vector going to these other organs. And if you look at the difference in the y-axis here, you have about 100 times more vector going to the liver than you have going to the brain. This is an approach that is dose responsive. It works to some extent, but it's definitely not very targeted. But nonetheless, we and others kind of explored how this approach can translate from rodents to non-human primates, and it does. You can achieve the vector will cross the blood-brain barrier in monkeys. I'd say our findings on this was that it was less efficient in monkeys as it was in rodents, but it does still work. But again, looking at the big picture, you have sort of some logistical barriers where you have high doses that need to be administered. You have very high delivery to other peripheral organs. It's not very targeted to the brain, and about a third of humans will have anti-AAV neutralizing antibodies in their blood, because AAV is sort of a naturally occurring virus. It circulates in humans, and a certain percentage of the population will already have some kind of immunity against the AAV9 capsid. And so basically, if you've been exposed to AAV in the wild and have these neutralizing antibodies, then this approach would not work. But with all those challenges, this approach was applied to spinal muscular atrophy, and this became the second FDA-approved AAV product. And I think many of you might be familiar with this story. And now there's this marketed drug called Zolgensma that carries the SMN1 gene that's missing in infants with spinal muscular atrophy. And the efficacy on this has been quite astounding. And I think to date, there's been about 3,000 babies, SMA babies, that have received this drug. So, so we're sort of in a situation where, you can inject AAV9 IV and you kind of get where it goes everywhere. And so the question is, can we target it more efficiently to the nervous system? And some of this could be modifying the virus. And there's a lot of exciting work happening here to try to make sort of different versions or flavors of AAV that are more targeted to the nervous system. Or the simple thing would be to modify the route of administration. And that's really what I'm going to go into here. That by simply injecting this vector into the spinal fluid, then this is a way to sort of concentrate the vector around the nervous system. And this is an approach that can be done kind of as an outpatient procedure in humans. It's much more challenging to do in mice, but at least we've gotten pretty good at it. And if you do this in mice, and just do a lumbar puncture and inject an AAV9 GFP vector, then you'll still get this sort of widespread distribution throughout the nervous system. And there's a couple things that I want to really emphasize with this. This is all published study, but this is kind of a, approximately a maximum feasible dose. It's getting, a pretty high dose of the vector and kind of reaching volume limits in terms of what would be a typical injection. So if you look throughout the brain parenchyma, you'll see that we're still getting pretty widespread distribution of the gene expression. If we zoom into a region of the brain, one thing to note is that we do see scattered cells, and these sort of dark punctate cells would be neurons, and these more bushy cells are going to be glia. So it's a mixture of neurons and glia. But it's definitely not every cell. And if we attempt to quantify this by looking at the copies of the transgene per mouse genome, we're averaging, about 5 to 20% of cells, in this study. So we're definitely targeting a minority of cells across the brain. We get better coverage in the spinal cord, where we're getting, 1 to 2 copies of the gene per cell on average. And it does go to peripheral nerves. And, so that's the one point I want people to take away from this is across the brain, we're just targeting a percentage of cells. And this is sort of the best that this technology can do right now. The other point to make is that if we look at peripheral organs, that the vector is still going out and escaping and getting into the periphery. And so we're still seeing vector going to the liver and to the heart and the muscle and kidney and other organs. So from a intra-CSF injection, we are getting distribution across the whole body to some extent, but now instead of getting 100 times more vector into the liver, now we're getting closer to a 1-to-1 ratio of vector going to peripheral organs versus nervous system. So this is all in mice. We did studies very early, over 10 years ago, exploring this approach in pigs, and these were about 20 kilogram pigs, modeling sort of small child. And so you'll see still pretty widespread distribution of the vector, throughout the hindbrain of this pig in this example. And then taking this forward into non-human primates, the overall conclusion was that the results that we got in mice translated pretty evenly going into non-human primates. And we concluded that there were some advantages over an IV administration because you could use a lower overall dose. If we're injecting into spinal fluid, we're actually avoiding those AAV neutralizing antibodies in the blood. And now we have, like I said, sort of reduced peripheral organ biodistribution. So kind of solving or improving on a lot of these problems just by changing how we inject the virus. Now, in terms of the degree of spread, since then we've done a few other studies really looking in non-human primates and taking the dose very high. And this is equivalent to sort of the highest dose levels that we're doing in humans right now. And this question always comes up about, how good will the distribution be when you move up to a larger brain and a larger animal? And at least what we've seen and what we've concluded is that we still see pretty good translation, that what we see in mice translates to what we see in large animals. And so this is just a biodistribution analysis this is 4 monkeys. And so, the 2 green dots would be the lower dose, and then the 2 blue dots for each bar would be the higher dose. And so basically we're still sitting on this sort of, 0.05 to 0.2 vector genomes per cell on average, although we did have one high performing monkey that seemed to have much better distribution. And we looked across 22 different brain regions, and this is all published recently, earlier this year, so you can look this up in more detail. But we still saw pretty even distribution across these different regions of the brain, whether they were sort of superficial, close to the CSF, or whether they were deeper brain structures. And similar to what we saw in mice, we still see the vector going to peripheral organs. And just to visualize this a little bit, this is another study that we did in non-human primates. This is only 2 animals, but it was for CLN7, was the transgene, and another vector that we have in human trials. So this was, in this case, we injected these into juvenile non-human primates at doses equivalent to sort of the maximum dose that we're using in patients. And this is just, but here we're visualizing the transgene expression using RNAscope. And so this is just sort of representative results where I took a slab of the brain that was kind of in the middle of the brain, and then pick some regions that are deeper in the brain parenchyma, and just showing here in these zoomed in panels that the red spots are actually staining for transgene RNA that's being expressed. And so we can see, again, what we expected from mice is we're definitely not transducing every cell. We're still transducing a minority of cells, but we are seeing it all throughout the different brain structures. So, what I want to sort of emphasize is that, this is kind of a platform approach here where we're demonstrating the ability to deliver some nucleic acid broadly across the brain. That at this point, we've tested this in mice, rats, pigs, dogs, cats, non-human primates, sheep, and then there's ongoing human trials for a number of diseases. So thinking about this as a platform approach, we started this, and I'm going to briefly talk about, sort of why we started this. And it was really inspired and initially funded around this very rare disease called giant axonal neuropathy. And so this is a very debilitating childhood neuropathy. It's ultimately fatal by about 20 years old. And it's a single gene autosomal recessive disease where they're missing a gene called gigaxonin. And this starts with sort of clumsiness as a toddler, inability to walk by about 10 years old, and then getting into the later teenage years, requiring different measures of increasing life support, reduced use of arms and hands, and it's basically due to the by missing this one protein, inability to process and organize intermediate filaments properly. So these form these intermediate filament aggregates that tend to create these bulges in axons, and it's essentially a length-dependent axonopathy where you get dying back of axons that eventually leads to neuron death. So not to go through all the preclinical data on this, because we published all of this about 5 years ago. We developed a gene therapy approach for this, kind of using this intrathecal AAV9 approach. We were able to improve the pathology of a mouse model and also improve its motor dysfunctions or its motor phenotypes. And then importantly, we tested this in a large GLP studies in mice, rats, and non-human primates, and saw absolutely no safety concerns. And so this was something that we took into a clinical trial. It was funded 100% from this, sort of grassroots patient organization called Hannah's Hope Fund. And the timeline for this was we started in 2008, before this technology with AAV9 even existed. And so we kind of developed this gene delivery approach to the nervous system, got proof of concept results in patient fibroblasts in an animal model, went to the FDA in 2012, early 2012, to kind of propose this intrathecal gene therapy trial that, to my knowledge, was the first time anybody had proposed to do that. We got good feedback from the FDA, and that led us through doing pharmacokinetics, pharmacodynamic studies, toxicology, clinical trial design, really kind of charting a path here, going through the alphabet soup of regulatory agencies. And then in 2015, we started a phase 1 clinical trial at the National Institute of Health. And I want to take a minute to acknowledge, really, this was a partnership with the NIH and NINDS, and the PI of that trial was Karsten Bonneman, who's been an amazing colleague to work with on this. And I want to just sort of celebrate a milestone here that's almost 10 years old now. But in May of 2015, we did the first in-human intrathecal gene transfer trial to broadly treat a CNS disorder. And this was, as far as we know, for any disease, the first time this has been done for any disease. And it was for this rare disease that I think we're up to, we sit around 100 patients in the world that we know about. But it was really due to the work of Lori Sames, who is a patient advocate, mom, Karsten and Diana on the clinical team. And briefly, we're working to get the long-term results of this clinical trial published, but we're sitting at this point where 14 patients were treated in this clinical trial. Follow-up is now, over 5 years for many of these patients. And we had the advantage that, really about 50 of the patients worldwide participated in a natural history study that was running parallel to the clinical trial, and kind of established a good, pretty robust data in terms of the natural history of the disease and the slope of decline. And one of the measures that they focused on that ended up being pretty predictive of the disease progression is this motor function measures 32 scale. And this showed a pretty consistent slope of decline of about 8 points per year. And so, you could compare patients to that natural history slope. And so one thing would be to compare them to natural history. And then we have our patients treated across different dose levels, and there, kind of appears to be this leveling off of disease progression. So that's one way to analyze this is against natural history. The other way would be to compare many of these patients participate in the natural history study. And so we have kind of lead-in data from these patients, and we can kind of compare each patient, what their slope of decline was before treatment, what their slope of decline was after treatment. And so, when we look at the individual patients, the data can be kind of messy. But if we combine all of this and bring in Bayesian statistics, then which are really amenable to looking at small patient populations in a clinical trial like this, then we see this sort of leveling off of disease progression post-treatment for these patients. And even though it's relatively small n's, these results were highly significant. Whether you compared patients against the natural history or whether you compared them to their lead-in data, either way, it was highly significant in terms of treatment effect. And the other thing that's I think ended up being fairly remarkable is some of this was these functional measures like MFM32, but there's other measures that were incorporated into this clinical trial that are non-subjective. One of those was looking at nerve conduction velocity and nerve conduction amplitude. And so, like I said, this is a peripheral, it's a central and peripheral neuropathy. You have this dying back process of the peripheral nerves. And so you can see that, these patients show a very sharp decline in peripheral nerve function. Some of them are bottomed out, where they can't actually detect any nerve conduction. And then post-treatment, we see, patients stabilizing, or many of the patients actually showing improved or restoration of nerve conduction post-treatment. And these are just two different sensory nerves that are in the hands, median and ulnar nerves. And so we see this in both. And, one of the safety concerns associated with intrathecal administration of AAV9 is, there has been some evidence that you can get some damage to dorsal root ganglia, which are these pockets of sensory neurons that sit alongside the spinal cord. And so there's a hypothetical risk that you could have the development of, some reduced peripheral nerve sensory function. But in the case of this clinical trial, we actually saw the opposite, where there is disease pathology in those neurons, and we were able to improve their function. So, how does this happen for such a rare disease? And, and this is, really where it gets kind of tough when you talk about the rare disease space, that I think most of the people in this meeting kind of recognize, is it really takes very strong patient advocacy. And, I want to acknowledge Lori Sames, who was, fighter mom that really drove all of this and pushed everybody to develop this treatment and keep carrying it forward. But, she also had a committed, supportive community of volunteers that, among other things, raised a lot of money. And, I think that they'll typically say they raised about $6 million in 6 years. And frankly, I think it was exhausting. It was amazing, but it was exhausting. And, again, I think as a lot of people on this call recognize, unfortunately, this is sort of how things happen oftentimes in the rare disease space. So what are the magic ingredients? I'd say, from my perspective, just speaking from my experience, I think it's important to link scientists and families. It's important to have compassion. We really have to care about making the lives of these kids better. You have to know what you're doing, and accountability comes of knowing that patients are really suffering and need something right now, and there has to be that recognition of accountability that every day has to be spent efficiently and as productively as possible. And like I said, all of this takes money. These types of treatments are very expensive to develop. So I want to go into sort of, again, this idea of this being a platform approach, and, how can we apply this to other diseases? And I'll give maybe one or two examples here. If we do have a situation where we deliver a gene to the brain and we're treating a minority of cells, we can point to examples like GAN, where treating that number of cells still translates into quite a meaningful benefit in the absence of a quote unquote cure. But there are a class of diseases where you can treat one cell and then it will express a protein, and then that protein is shared with neighboring cells, and that's called cross-correction. And so there's many lysosomal storage diseases that sort of have this mechanism of cross-correction where, if you treated 5% of cells, then those 5% could become factories to produce the gene and share it with all of the neighboring cells. And an example of this is Krabbe disease. This is a very, very devastating leukodystrophy. It's fatal typically by about 1 to 3 years old in the typical presentation. Autosomal recessive disease, missing this galactocerebrosidase gene, and the only, if I can say it, the only notoriety around this disease is that Jim Kelly, he's a Hall of Fame quarterback for the Buffalo Bills. His son Hunter died of this disease, and they've done an amazing job of advocacy to raise awareness for Krabbe disease. We did a pretty large studies in mice that model Krabbe disease, but there's also a naturally occurring dog model of Krabbe disease that's maintained at University of Pennsylvania. So we worked with Charles Veit and Allison Bradbury at University of Pennsylvania to run a gene therapy rescue study in these dogs. And in short, if we just look at survival curve, untreated dogs end up having to be euthanized about 16 weeks old, about 4 months. And so if we treated dogs very early at 2 weeks old and with kind of our highest dose, then, we published this in 2020, but, the dogs went out about 2.5 years with all the treated dogs that got the high dose at the early age did quite well. But if we treated them with a lower dose, or if we injected them older at 6 weeks old, then we saw much less impressive efficacy results. And I'll just highlight here, let's see if these videos will show. I hope that these don't get kind of choppy over Zoom. But this is kind of what untreated dogs look like at 10 weeks old, where you can see that, where they're starting to have, the neurological symptoms really setting in with difficulty walking. And but then we have, a number of treated dogs that were given a high dose in an early age. And this is just a video taken at 1.5 years old where these dogs had no clinical symptoms at all. This was essentially a complete rescue. So this is one of those diseases that we have in the lab that we're actively trying to move into a clinical trial. But it's kind of faced with the same barriers that everything else does of trying to get the funds and trying to go through the process. So we have been working on SIRF1 related Leigh syndrome, and so we published an initial study on efficacy in a mouse model. And I'm not really going to go into this, but I'll highlight that Dr. Qingyuan Ling will be speaking at 11:15 Central Standard Time, kind of giving an update of where we stand with SIRF1 gene therapy. And thinking about this as a platform approach, this is a short list of, not everything that my lab works on, but most of the things. And in Casey's introduction, she mentioned, we have 7 different diseases that we've moved into clinical trials. Really, of these, the GM2 gangliosidosis, Tay-Sachs and Sandhoff, CLN1 Batten disease, and CLN5 Batten disease. Those 3 benefit from cross-correction. But these other ones don't. And so we still got, the amount of gene transfer that we were getting was still enough to provide a pretty meaningful benefit, at least in the animal models of disease. And I think in GAN now we have long-term data in humans showing a clear treatment effect. So in the bigger picture, again, thinking about this as kind of a platform approach, there's over 24 clinical trials now that are using similar approaches of AAV9 or similar vectors that are injected into the cerebral spinal fluid. And so this is a recent review that we published in 2023 that kind of outlines all this. So, I guess the question is, so if we have the technology to do this, we're applying it to a handful of diseases, then where do we go from here? And, I think I probably don't need to go through this too much for this audience, but we talk about rare diseases. It's a large but hidden problem. Over 10,000 identified rare diseases, about 8% of the population. 80% of these diseases are genetic. So in many cases, gene therapy, at least conceptually, makes sense. 50% of these patients are children. And then it gets really difficult from here on out because 30% of those children don't live to see their 5th birthday. And very few of these have FDA-approved treatments. And so I think that this right here is enough. The unmet need is enough of a reason to be trying to solve this problem. But if we look even at the economic burden of this, this was a study published by the Every Life Foundation in 2021. They kind of estimated the total economic burden of rare disease in the US is close to $1 trillion a year. So there's a profound unmet need for these patients, but there's also strong economic incentives to try to figure this out. And so, how do you tackle, well, now we're up to about 10,000 rare diseases that affect a significant amount of Americans and people around the world at some point in their lives. And I think up until now, the answer has been one disease at a time. How do you eat an elephant one bite at a time? How do you solve rare diseases one disease at a time? And that's a little bit unsatisfactory. So, the idea here is to make standardized platform approaches that can be readily applied and kind of break down the barriers of developing these sort of individual disease treatments. And one of the things that we're very excited to be participating in is this bespoke gene therapy consortium that's being spearheaded by the Foundation for the NIH. And this is estimated about a $97 million effort of financial and in-kind commitments from, basically this consortium of different NIH institutes, life science companies, and nonprofits that are all coming together in sort of pre-competitive consortium to standardize this approach to gene therapy for rare diseases. And so hopefully the idea here is to drive forward treatments for, they selected 8 diseases, to drive forward treatments for those 8 diseases, move these into clinical trials, and then basically make all of that information public. So and try to organize it into a public playbook that other groups could follow on, how to do all the preclinical studies, how to do the manufacturing, how to do the tox studies, how to put together the IND, and how to do aspects of the clinical trial. And so of those 8 diseases that were selected to be driven forward in this consortium, there were 3 neurological diseases. And so those 3 diseases were actually all developed out of my lab. The MSD and CMT4J were done in collaboration with Jackson Laboratories, but we're excited to be working to move these forward and hopefully kind of continue our efforts to make all the information around these public to help others do the same thing. So, what's next? Like I said, the technology is what it is right now, but it's still not perfect. So we have kind of the status quo right now of where the technology is that AAV9 and other vectors that are kind of similar to AAV9 can be administered IV or intrathecally. Actually, both will work if you're trying to target the brain. But both of these approaches have been validated across numerous labs for effective disease-modifying gene therapy or gene transfer to the nervous system. And like I said, this has been across many different small and large animal models and also humans. But I do want to stress, there's significant limitations with this technology. We're still targeting a minority of cells in the brain. So let's think about treatments, and not necessarily cures most of the time. And so it's also, we kind of have to go disease by disease to try to assess in preclinical models, does that level of gene transfer, does it confer a transformative benefit, or is it not quite good enough yet? The other thing that I really didn't talk about is that AAV gene therapy is still limited to one dosing event. So if patients have a sort of low level of naturally occurring antibodies in the blood, then we can avoid those and dose those patients by injecting into the spinal fluid. However, we know from experience in the clinical trials that once we dose a patient with very high doses of AAV, then they mount a very strong antibody response, and we effectively immunize those patients against another dose. Those antibody levels get quite high. So although there is quite a lot of work trying to figure out how to block those antibody responses and permit redosing, I think right now we have to assume that this is a one-time dosing event. So, the take home is that this is not a one-size-fits-all, but we hope that it's a one-size-fits-many and that the technology will continue to improve. And the, sort of the gaps are, we really need better capsids. I think that we're making, we're making a lot of progress with AAV9, but we need to have something better to get sort of, greater efficiency and start treating the majority of cells across the brain. And also, allow us to use lower doses. We need better control of adverse immune responses. This is really where the problems have happened in the clinical trials, especially when very high doses of AAV are being used. And right now, the biggest barrier for these treatments to move forward is around manufacturing, because the manufacturing is very expensive. And so if we can, get to where we can make more AAV, kind of with less, then it'll make these things a lot easier. And oftentimes we end up with situations where too much expression of the gene can be toxic, and so the whole field needs to be looking at improved gene regulation strategies. And kind of outside of the lab, I'll say that we really, really need some new policies and public health initiatives to make it easier to develop and translate these rare disease treatments. Because, I think we've probably all experienced this with diseases as rare as Leigh syndrome. It's often very difficult to attract pharmaceutical companies, and the system as it is, it's very hard to fund and develop these treatments towards approved products outside of a typical sort of industry-led drug development paradigm. And but even though I think a lot of these treatments, if we could have them make sense from a public health perspective, we just we don't really have a mechanism to make it happen. So with that, I want to acknowledge my team that comes to work and works very hard every day to try to help us develop and move forward these treatments, and also lots and lots of different funders and collaborators that have helped us out throughout the years. So with that, I want to say thank you again for the opportunity to talk, and happy to answer any questions with the time that we have left.
As always, super impressed. Thank you for the gene therapy 101. I always break down and cry when I see that dog video, and I know the video was kind of lagging, and it's just amazing to see, these little puppies who are barely able to walk, and then after their treatment are running around, just you can't even tell what's going on. It's just amazing, and thank you for your work. We do have some questions, so I will go to the Q&A box right now. Let's see. We will start with this is from Volkmar, would non-viral transinfection be a valuable alternative, using, for example, LNPs to avoid the immune response?
Yeah, no, it's a good question. And, right now, any of the sort of non-viral vectors, I think, we've had success delivering to the muscle, like the COVID vaccines. There's been success delivering to the liver and other localized targets. But in terms of getting widespread distribution across the brain, I'm not aware of any technology that really reaches the level of efficiency and widespread distribution that AAV can do. I think antisense oligonucleotides can get pretty widespread delivery, but those aren't necessarily nanoparticles.
Great. Next question, Nikolai. He is from Romania. So thank you for joining all the way from Romania. His daughter has ATP6 Leigh syndrome. And he says, considering a successful delivery of a healthy gene in a cell, would that gene replicate upon the cell division process, thus multiplying the treatment effect? I guess that question also relates to how long the treatment would last.
Right. Yeah. And so this is an important limitation of AAV. So when AAV delivers its genome, then that genome is basically converted to an episome, or like a mini chromosome. But it does not contain the elements that the cell would use to recognize it, to divide and copy whenever the cell divides. So in short, if you deliver AAV to a dividing cell population, then the AAV genome will get diluted out over time. But if you're in a post-mitotic cell like a neuron, then the genome should be stable. And theoretically, it should be there for life. And so that's where the nervous system ends up being a good target, because your neurons they're post-mitotic, they're permanent. The ones that you have, in like late childhood are the ones that you have when you die. Yeah, but if you're trying to target like skin or mucosal membranes or other cell populations that are rapidly dividing, then AAV is really not a good vector.
Okay, thank you. This question is from Francis. You did mention ASO, and ASOs are not for loss of function phenotype, correct?
Yeah, I think you want to be careful about generalizing like that. So ASOs are typically used to like knock down a gene. If it's a gene that's sort of toxic, or the mutation is toxic, then you could knock it down. There are some strategies that people are trying to use with ASOs to like upregulate, like if you have a haploinsufficiency or something where you just aren't getting enough expression, of trying to use creative strategies to use ASOs to basically make the healthy or functional copy of the gene express a little bit more. But generally speaking, ASOs are for sort of loss of function, or are to knock down sort of toxic alleles.
I hope that helped, Francis. We are at time right now, so Dr. Gray, you do have several questions in the Q&A. If you do have time, can you run through them and kind of answer those just by typing an answer? That would be greatly, greatly appreciated. We know that you're busy, so thank you so much for your time. And as always, we're so thankful to be working with you on Leigh syndrome.
Yeah, thanks again for the invitation. This is a real pleasure.
Okay, you've got 10 questions in the Q&A.